Hi, everyone. I'm Richard Naylor from the Friends of the Pine Bush, and we are once again honored to co-sponsor with Commission staff our monthly science lecture, Recovery Efforts for the Endangered Karner Blue Butterfly at the Albany Pine Bush Preserve. While we can't meet in person, we do feel fortunate to have a social media platform like Zoom to still have the program. One of the neat features of Zoom is a question and answer or Q&A for questions that occur to you during the presentation. And you can use that by clicking Q&A at the bottom of the screen and then type your question. The speaker and Dylan, who's the moderator, can see the questions and answer at the end according to the question. If you have a laptop or a desktop, you might need to hover your mouse over the bottom of the screen. And for an iPad, you probably will have to swipe up. Before Dylan introduces our speaker today, let me tell you about next month's science lecture webinar. It's called Plains of Glacial Albany. And sand, of course, is the basis here of why we have a pine bush preserve. The program will be on Thursday, August 20th, starting at 7 p.m. Our speaker is Alvin Bryce, who first explored the Albany Pine Bush as a graduate student, studying plant ecology at SUNY Albany, which is now the University at Albany. That was in 1967. Over the past 50 plus years, he has gotten to know many of the people trying to protect this unique ecosystem and also those conducting detailed studies on the flora, fauna, and natural communities of the Albany Pine Bush. But now back to today and Dylan. Dylan.
All right, thank you so much, Richard. And thank you so much to the Friends for regularly sponsoring our science lecture series. It's a really great partnership. So my name is Dylan. I'm the field ecologist and entomologist at the Albany Pine Bush Preserve, and it's my great pleasure tonight to introduce my coworker, the conservation biologist at the Albany Pine Bush Preserve, Dr. Steve Campbell. So just to give you a little bit of background, he's went through a lot of schooling. He got his Bachelor's of Science from the State University of New York, College of Environmental Science and Forestry, and that was in environmental and forest biology. He then went to Mexico State University for his Master's of Science degree in biology and experimental statistics, and also earned a PhD at New Mexico State University in wildlife ecology from the University of Maine. He's done research with birds, looking at plant hybrid zones and willow biomass projects. He's also worked with the Chittenango ovate amber snail at Chittenango Falls in New York. And he's taught undergraduate and graduate courses in wildlife ecology, statistics, and population ecology. While he was a visiting assistant professor at Eastern Kentucky University. He's now teaching undergraduate classes at SUNY Albany. And he's joined us in, I think, 2012, right, Steve? 2012 here at the Albany Pine Bush Preserve as our conservation biologist. And whereas I kind of run the field data collection side of things, Steve runs data management. So he designs a lot of our experiments and our research, and he analyzes that data. And he's presenting to us tonight on our data from our recovery efforts for the Karner blue butterfly. So with that, I will hand it over to Steve.
All right. Thank you, Dylan. And thank you all for tuning in and supporting our science lecture series. I think since I've been here, since I've been working here, the attendance at our science lecture series has grown considerably. I think when I first gave an update on our Karner blue recovery efforts in 2013, there were only about 6 people in the audience. And I think today we have almost 60 people signed in remotely. So I'd like to thank you for showing your support and for taking an interest in our work at the Albany Pine Bush Preserve. Okay, so I'm just going to jump right in and introduce the star of the show. I know you're not here to see me, you're here to hear about the Karner blue. So if you've seen the Karner blue, or even if you haven't, then you know it's not a very big butterfly. It has a wingspan of about 2.5 centimeters or 1 inch. So with its wings closed, it's really not that much bigger than your thumbnail. All individuals are grayish on the underside of the wings with orange crescents near the margins. However, on the dorsal side of the wings, they are sexually dichromatic, which just means that the males and females are different colors. Males tend to be a deeper, richer blue, and the females tend to be a duller blue, and they have these orange crescents on the trailing edge of their hind wings. Historically, Karner blues occurred throughout oak savanna and pine barrens ecosystems. The oak savannas occur in the Midwest, south of the Great Lakes, and the pine barrens ecosystems occur in the Northeast. And these ecosystems are largely open areas. They're sort of these shrubby grasslands with relatively few trees. Humans have destroyed much of these ecosystems, so currently butterflies are really limited to the remnant patches of these ecosystems. Incidentally, however, human activity has also created habitat for this species. By cutting forests or maintaining open areas for power line right-of-ways or airports, humans have created vegetation communities that are really structurally similar to those that they have historically used. However, it's important to note that just having these open shrubby, grassy conditions isn't all that is needed. An essential component of their habitat is wild lupin. The larvae are dependent upon this plant. While the adults will feed on the nectar of many different species, the larvae will only feed on the leaves of wild lupin. So if there's no lupin, you're not going to have any Karner blues. Here's a typical year for the Karner blue butterfly. They overwinter in the egg stage, and then in spring, the eggs hatch out probably sometime around early May. The larvae feed on wild lupin, they grow, they pupate, and then the first brood of adults begins to emerge at the end of May and then through June. Those adults live about 3 to 5 days is all, and during which time they find a mate, they lay eggs, and then those eggs develop, and then they hatch, and then those larvae feeds on lupin, and then they pupate, and then they give rise to a second brood of adults that emerge through July into sometimes in early August. And that completes the annual life cycle for the species. And this is what's known as a bivoltine life cycle. So that's just, it has 2 generations per year. And the graph here is just sort of a schematic of the adult abundance over time. So the first hump here represents the first brood. Second hump in abundance here represents the second brood. Okay, so why is a Karner blue endangered? Well, the threats to the Karner blue are really the same that affect most threatened endangered species. Much of its habitat has been destroyed and replaced with human development. Here are a couple local examples that you may recognize. Up here at the top is Cross Cape Malls. Down here is the University of Albany. Both of these were built in places that were historically pine bush. Habitat fragmentation usually accompanies habitat loss. And habitat fragmentation makes it difficult for individuals to move among the remnant habitat patches. And so for the Karner blue, even landscape features such as roads or even just strips of forest can act as fragmenting features. Habitat degradation is also a problem. If the process that keeps these areas open are disruptive, these areas can become overgrown with trees and shrubs, leading to forest-like conditions, which are really not suitable for the Karner blue. So for example, if fire is allowed to periodically burn, it would slower prevent the establishment of trees and shrubs, as you can see up top here. However, if you suppress the fire, that is going to allow trees and shrubs to become established at a greater rate than they would, that would happen if fire was allowed to periodically burn. Invasive species also pose a threat, especially invasive plants. These invasive plants can shade out lupin and other understory plants needed by Karners. For example, here you can see this is in the spring, the honeysuckle is starting to leaf out. And what can happen is the honeysuckle can take over the understory and shade out a lot of the other native plants. Or you can have something more severe that can come in and completely alter the vegetation structure and composition into something that's completely unsuitable like a forest. And so this is what happens if black locust takes hold. It basically turns it into forest-like conditions. Unusual weather events can also pose a threat, especially if the population is already small. For example, if you have a spring or summer drought, that can stress the lupin, which then could reduce larval populations. Or you can have cold, wet weather during while the Karners are supposed to be flying. And then really that can do is can reduce the amount of time that's available for them to mate and lay eggs. remember, they only live for a few days. So if you have a string of a few days of bad weather, that can really possibly prevent some of these individuals from reproducing before they die. And then over the long term, climate change is also likely to pose a threat. I'll get into that a little bit more towards the end of the talk, but it is a looming threat for the species. So all of those threats have acted together to lead to a greater than 90% decline in abundance and distribution of this species since the 1970s. This yellowish tan area is where the species used to occur. It used to occur throughout the Northeast and the northern Midwestern states. Now natural populations are limited to Wisconsin, Michigan, and New York. The population in New York is the only natural one left east of the Great Lakes. There is a population in New Hampshire. I don't consider it naturally occurring because the population did actually go extinct, and it has since been restarted with captive individuals from New York. But there is a population in New Hampshire as well. And I should mention that none of these populations are considered viable, meaning if nothing is done to help these populations, they stand a high likelihood of going extinct. So to help prevent the further decline of this species, the U.S. Fish and Wildlife Service listed as endangered across its range in 1992. Now, when a species is listed under the Endangered Species Act, a recovery plan must be developed, and that recovery plan outlines actions that will be taken to recover the species. And also included within that plan are criteria by which recovery will be determined. And so for the Karner blue, in order for the Karner blue to be considered recovered, there must be one or more viable population in all 13 of the recovery units. So these stippled areas that you see here are recovery units. And in the Glacial Lake Albany recovery unit where we are, they're calling for 3 viable populations in the Glacial Lake Albany Federal Recovery Unit. And what I mean by a viable population, for a population to be considered viable, there has to be at least 3,000 first or second brood adults in 4 to 5 years. Now, New York has its own listing process, and New York State Department of Environmental Conservation listed the Karner blue as endangered in the state in 1977. So within the Glacial Lake Albany Federal Recovery Unit, there are 4 New York State recovery units. And for New York State, DEC wants viable populations in all 4 of these state recovery units. And the definition of a viable population in New York State is the same one that they've adopted at the federal level. And again, that's 3,000 first or second brood adults in 4 to 5 years. Okay, so now let's turn our attention to this particular state recovery unit, the Albany Pine Bush Recovery Unit. And let's start with just the general question, if you're not familiar, what is the Albany Pine Bush? And the Albany Pine Bush is just a local name for an ecosystem that's known as the Northeastern Interior Pine Barrens. And this ecosystem at its base is a sand dune field. If the vegetation were removed, it would look like the blowing sand dunes of the Sahara Desert. So here you can see an area where the vegetation has been removed as part of the restoration, and you can see it's just nothing but sand. And because the soil is largely sand, the soil conditions are infertile and it's acidic. And not a lot of plants can tolerate those conditions. And on top of that, because the water drains quickly through the soil surface dries very quickly and it makes the area prone to burning. So plants that historically occurred here had to be adapted to these dry, infertile, acidic soils, and they had to be able to survive periodic fire. And the historic plant community was dominated by pitch pine trees, as you can see here, scattered around in the canopy. And then it was an understory of scrub oak and heath in the shrub layers, and then with grasses mixed in. And hopefully if you look at this picture of the pine bush, you recognize that this is dramatically different than the deciduous forest that surrounds the pine barrens and occurs throughout much of the Northeast. Now this Northeastern Interior Pine Barrens is far from a homogeneous ecosystem. There are different ecological communities that make up the ecosystem. There are successional northern sandplain grasslands. These are communities that are predominantly grasslands with very little tree and shrub cover. There are pitch pine scrub oak barrens. These have scattered pitch pine trees in the canopy and then scattered patches of scrub oak, heath, and grassland in the understory. There are pitch pine scrub oak thickets. These are really similar to the barrens, but there's a much greater scrub oak coverage and there's less grass in the understory. And then there's pitch pine oak forest. And this is closer to a closed canopy forest of pitch pine. There is some scrub oak in the understory and there's a relatively sparse herbaceous layer. And these can all really be considered as successional variants of each other, meaning that these communities can transition from one to another depending on the frequency and intensity of disturbance. So for example, if disturbance is frequent enough, or sorry, if it's infrequent enough, then the scrub oak or pitch pine, the scrub oak or the pitch pine, scrub oaks or pitch pines in the barrens could become dominant and turn a pitch pine scrub oak barrens into a thicket or a pitch pine oak forest. However, if disturbance is frequent enough or intense enough, it could revert any of these back to grassland. If they burn hot enough and frequently enough, these could all be converted into grasses. So we consider some combination of these four communities to be representative of the historic vegetation communities at the pine bush. So these communities, especially pitch pine scrub oak barrens, are our main conservation priorities. There are other ecological communities out there. There are pine barrens, vernal ponds, red maple hardwood swamps, and shallow emergent marshes. These are all communities that we also want to conserve. They were historically part of the landscape because fire was naturally excluded from these areas because they were so wet. There are also other communities out there such as Appalachian oak pine forest, northern successional hardwood forest, and southern successional hardwood forest. These are communities that were unlikely to have occurred in the pine bush historically. Periodic fires would have prevented them from becoming established. We have them now because we've had decades of fire suppression. But these are communities we do not want. And I'm going to come back to how we're dealing with these in a little bit. Okay, so now you know what it is, why is it important, why do we care about the Albany Pine Bush, what makes it special? Well, two of the communities I just mentioned are globally rare. One is the pitch pine scrub oak barrens. New York State Natural Heritage Programs gives that a G2 S1 rating. The G2 rating means that there are less than 20 occurrences of this ecological community in the entire world. And the S1 means that there's fewer than 5 occurrences of this community type in New York State. And I should mention that the Albany Pine Bush is considered one of the best examples of this community remaining in the world. The other community is the Pine Barrens Vernal Pond. Because these are embedded within Pine Barrens, these are also globally rare. Natural Heritage gives them a G3, G4, S2 rating. Also, another reason why they're important is not surprisingly, the unique plant communities of the pine bush support a unique assemblage of animals that are really quite distinct from those communities that you're going to see in the more common deciduous forest. And many of the animals that occur here are of conservation concern. For example, there's 78 species of greatest conservation need, wildlife species of greatest conservation need, that have been documented here in the preserve since 1980. Obviously, one of the best known examples is the Karner blue. There's also the eastern spadefoot, the brown thrasher, inland barrens buck moth, prairie warbler, and eastern hognose snake. But you don't just have to take my word for it. Its importance has been formally recognized by New York State DEC. It's been recognized in its bird conservation area. National Audubon Society has also recognized us as an important bird area. And most recently, the National Park Service has recognized us as a national natural landmark due to the rare geology and ecology of the pine bush. Okay, so now let's turn our attention to the Albany Pine Bush Preserve. The preserve was established by an act of legislation in 1988 to protect the remnants of a northeastern interior pine barrens ecosystems and the globally rare inland pitch pine scrub oak barrens community that was within that ecosystem. So looking at this map of the preserve should make it clear that we're dealing with a very urban preserve. You can clearly see there's development all around the preserve and in between pieces of the preserve. It's located between the cities of Albany down here and Schenectady up here. I-90 runs right through the middle of it. I-87 cuts through here. We have a landfill, we have shopping malls, railroad tracks. Here, neighborhoods and businesses, golf courses, neighborhoods and businesses all around and in between pieces of the preserve. So this should give you some idea of what we're up against in protecting this ecosystem from an ecological standpoint, but also keep in mind the social and political implications of this kind of situation. We have lots of neighbors, and much of what we do is highly visible to them. And as you will see, we do some pretty drastic things to the vegetation as part of our management efforts. And these are not always understood or even welcomed by our neighbors. And we also have an endangered species which presents its own sort of unique set of challenges. So in addition to dealing with the natural environment of this landscape, we also need to navigate the social and political environment as well. Okay, so now that you have a little bit of background about the species, the ecosystem, and the preserve, now let's talk about management strategies for recovering the Karner blue within the context I just provided. And in general, our management can be roughly divided into system and species-based approaches to management. For system-based management, we restore the ecosystem and then we reintroduce the processes that are going to maintain that ecosystem. So these actions should support and bring back many of the species associated with the ecosystem. You can think of this as a strategy to sort of, if you build it, they will come. If we set up the ecosystem, a lot of the species will then return to that ecosystem. However, system-based management may not be enough for all species. Some species may require more focal, more focused management actions, such as either captive breeding or provision of food resources. And these are really species-based approaches, and they're designed really to benefit only one or a few focal species. For the Karner blue, we use these types of management in combination. The system-based approach puts in place a functioning ecosystem, and then the species-based approach helps bolster the population size of the Karner blue. Okay, so in the next group of slides, I'm going to talk about some of our management efforts that fall under system-based management. And so keep in mind as I go through these slides that with system-based management, we're looking to restore the ecosystem, and then we want to reintroduce the processes that are going to then maintain that ecosystem. So one of the most important things we can do to restore ecosystem function is grow the size of suitable pitch pine scrub oak barrens vegetation that are protected. We want to grow the size of the preserve. And to this end, we are slowly acquiring land to add to the preserve. When the preserve was established, when it was founded in 1988, it was only about 800 hectares. We are now at about 1,300 hectares, and we're aiming for about 2,500 hectares in total. And I mentioned hectares. If you're not familiar with the metric system, a hectare is 100 meters by 100 meters. Or if you're going to make me put it in English units, it's 1 hectare is about 2.5 acres. And importantly, when we're trying to add land to the preserve, we are trying to connect areas of the preserve to reduce fragmentation and increase continuity among the protected areas. So a lot of what we're looking here to do is sort of fill in some of these blanks, and we're trying to do that with habitat that has some restoration potential. We obviously, if Crossgates was ever abandoned, I don't think that was something that we would ever try to restore back. I feel like that's probably too developed and too gone. So a lot of what we're looking to grow the preserve with is land that has high restoration potential, and so that's what you can see here that we're in the darker blue that we're looking into the future to try to acquire. Now, it's important to keep in mind that much of the land in the preserve and nearly all the land that we add to the preserve is not high-quality pitch pine scrub oak barrens. It's not high-quality pine barrens habitat. Much of the vegetation of those areas have been altered by years of fire suppression and invasive species. As such, much of the management we do with the preserve is aimed at restoring vegetation to its historical condition. So in particular, we spend quite a bit of time trying to control invasive species. Here are just a few examples of invasive species that we seek to either eliminate or control. There's black locust, oriental bittersweet, multifloral rose, and honeysuckle. So these are ones that have invaded, but also without natural disturbance such as fire, we also have some native species that have become overabundant. And for these, we're looking to decrease their abundance and distribution of these overabundant native species. Now, I don't have time to go into how we deal with all of these species individually, so I'm just going to cover a couple of our big ones. And so I'll start with black locust. This is sort of one of our more extreme examples. This is a black locust stand. We're in the inside of a black locust stand. The canopy is closed. The understory vegetation is completely altered from its historic condition. This is completely unsuitable for the Karner blues. And we decided the best way to deal with black locusts is really just to completely remove them. So we cut them down. We leave any residual, if there's any pitch pine or oak trees that have persisted among the canopy, we'll leave those. But otherwise, we cut down all the black locusts. We remove the logs. We rip up the stumps, and then we bury the stumps about 20 feet down. And when we do this, we're essentially starting from scratch. We're right back down to the sand. And then we come in and we plant that area with native plants. And after a few years of growth, we hope to end up with something like this. So with this type of restoration, we've essentially taken this from a southern successional hardwood forest, and we turned it into a successional northern sandplain grassland. Although this is still not pitch pine scrub oak barrens, we are at least now on this continuum. With either some time or some additional planting of scrub oak and other native woody shrubs, these grasslands will eventually become barrens. And with this map, this is just to show you our progress so far. The blue areas are the places where we've removed black locust. And we've removed about 150 hectares of black locust. And there's, I think, about the same number of hectares that's still remaining to be removed. So we still have about another 150 hectares of black locust that we still need to remove from the preserve. An example of an overabundant native species that we are dealing with is scrub oak. We want scrub oak in the understory. Obviously, if it's an inland pitch pine scrub oak barrens, it's an integral part of the community. But in this case, we have it's sort of too much of a good thing. The scrub oak can form dense covers that crowd out all the other plants. And so what we're interested in doing is thinning out the scrub oak. And we do this by first mowing the scrub oak, and then as it resprouts, we walk around. Well, usually we hire contractors to come in. We walk around and sort of randomly treat scrub oaks as they are starting to grow back. And what that does is it creates opening now for non-woody perennials. So here you can see the scrub oaks remaining, but now we've created all of these openings in between the scrub oaks. That allows other plants such as lupine and other native, other nectar species and other native grasses and stuff now to grow in these openings, and now we've created between the scrub oaks. So the goal of this management is to decrease the amount of thicket that we have in the preserve. So we want to decrease pitch pine scrub oak thicket, and then by thinning that out, we're essentially creating pitch pine scrub oak barrens from that. So simply thinning this out, maybe planting in some natives, or sometimes if there's natives in there, they'll be released. We go from thicket to barrens. And we've done this in these areas that you can see here in blue. We've done that in about 148 hectares of the pine bush. We've thinned out the scrub oak and opened up to allow in the other native plants. Okay, now remember, system-based management is not only about restoring the conditions, but also we want to reintroduce the processes that are going to maintain those conditions. And without bringing back those processes to maintain those conditions, the work towards restoration will essentially be wasted. Everything will just sort of revert back to what it was. So in this case, the process that maintains those conditions is fire, and we reintroduce fire through prescribed burns. We try to burn as much as we can each year, but being in an urban area really limits what we can do because we can only burn under very specific conditions. For example, conditions cannot be too wet, or they can't be humid, or they can't be too wet or humid because the fire will not carry. Or we can't have it too dry either because it will pose too much of an escape risk. And also, really importantly, the wind needs to be blowing in the right direction for any part of the preserve to be burned. We can't have smoke blowing across the thruway or into businesses or into people's yards. So we really need to be careful about, depending on where we're burning in the preserve, which direction the wind is blowing at that particular time when we're looking to burn. So we would like to burn as much as we can each year, but the reality is that we usually do not burn as much as we would like to because it is difficult to meet all of those conditions. Now, fire can be used as a restoration tool if you wanted to try to turn some of these communities from one to another. For example, if fire is frequent enough, and intense enough, we could turn pitch pine scrub oak forests into barrens or even into grasslands. However, we mostly use fire as a maintenance tool. So that means once we restore a particular community, we use fire to maintain that community. So for grasslands, that means we get out there and try to burn it every 2 to 3 years to keep out the woody vegetation. If we want to have it as a barrens, we really need to get in there every probably 5 to 10 years to prevent it from turning either into thicket or back into forest. And here, this shows you our footprint of our prescribed burns. These areas here in blue have been burned 1 or more times since 1991, since we started burning. So some of these areas have been hit multiple times. Some of them have only been burned once. Our goal is to burn about 80 hectares per year, but you can see that we've only reached those goals. So 80 hectares is about here. You can see we've only reached those goals in a handful of years. More so, we've reached those goals more recently because of our fire manager, Tyler Briggs, has been doing a really good job at getting fire on the ground. But it really still comes down to what conditions that we're presented with of whether or not we can burn. Okay, so that should give you some idea of how we're managing the ecosystem. Again, those management actions are aimed at getting the major components of the ecosystem restored and functioning. And doing that is going to bring back many of the plants and animals that are dependent upon the pine barrens. But now I want to turn to species-based management. So these are, again, they're activities that are focused primarily on just helping a single species, which in this case is the Karner blue. Now, obviously, if we want butterflies to use these areas that we've just restored, we need to make sure that we establish lupin in those areas where it does not exist. Or we want to supplement the population. If there is still lupin there, we want to supplement the population where it's low. So to do this, we collect lupin seeds, we dry them. As you can see here, this is our field ecologist and botanist standing in front of all this collected lupin seed, where they set it out to dry. And then when that seed is dried and prepared, we take them out and then we plant them into areas, either newly restored areas, or we interseed it into areas if we want to increase the population, the density of lupin. And then hopefully we put those out there, the lupin germinates, and we get lots and lots of lupin. Now, it's important to note that we're not after just wide open fields full of lupin. Lupin is necessary, but it's still not enough to ensure good Karner populations. We actually think the larvae do better in partial shade than if they're just sitting out in the full sun, where they have very little shelter and can potentially bake on really hot days. So we think here with some structure and shade provided by scrub oak and pitch pine in the canopy will provide some structural variation and some shading that will actually be better for the larvae. And we also want to make sure that there's plenty of other nectar species available. Although, again, the larvae require blue lupin, the adults will feed on nectar from flowers of many different species. And so we need to make sure that we have a variety of nectar species that differ in the time of years that they flower, so that we can ensure that we have nectar available over the spring and summer. We want it available across both broods. So this map shows the extent of the lupin in the preserve. So you can see here, it's really hard to see, but this purplish pink stuff, this is the area where the lupin existed prior, when the preserve was established. These are the only areas where the lupin existed. It's only about 4 hectares. So there's a patch here, a couple small patches here, one here, and I think one here, and maybe one here. So really not that many. But since then, you can see we've done a lot of effort here in these green areas. You can see that these are all areas that we've restored and that we've planted with lupin. So we've planted 290 hectares of lupin and other native species into the preserve. We usually don't just plant lupin. Usually there's a mix of species. But we've planted that mix and lupin in 290 hectares, which is considerably up the amount of area of lupin, again, from when the preserve was still first established back in 1988. Now, once we restore the patches, the butterflies just don't show up to them, at least not right away, especially if patches are isolated from other patches. They don't move very far, so butterflies might eventually make it to some of these isolated patches. But we don't necessarily want to wait that long. So what we did is we sort of, we accelerated the colonization of those patches, and we did that through captive rearing and release. So what we did there is we collect gravid females, so females that had eggs in them. We collected them on site, and then we transport them to the New Hampshire Department of Fish and Game. They had the facilities to rear the eggs. They tended the larvae, and then when those larvae pupated, they gave them back to us. So these are pupae. Now, at the beginning of the captive release program, between 2008 and 2010, the pupae were put out into the restored patches in these contraptions. And what this did is allowed these guys, when they came out of their pupae, they closed and they were able to fly directly out into the patches. Now, this setup worked well as long as conditions were not too hot. In years when conditions were very hot, we lost a bunch of pupae. They essentially cooked in the sun. So starting in 2011, we moved the operation indoors. And so when the pupae were ready to eclose, we'd put them in these butterfly tents with a nectar source. And then soon after they emerged, usually on the same day that they emerged, they were, here you can see Dylan walking them out and releasing them into a restored site. And so over here on the map, you can see these are all the places where we released butterflies into the preserve. And so from 2008 to 2015, we've released about 8,000 butterflies into 27 different patches within the preserve. And this graph shows our release efforts in each year. So the green bars show how many pupae we received from New Hampshire Department of Fish and Game. And the blue bar shows how many of those pupae successfully, how many of those butterflies successfully eclosed and then were released into the preserve. This vertical dashed line here, that just denotes when we shifted from allowing butterflies to eclose outside versus inside. So when they are outside, you can see if conditions were good, like they were in 2009, we had a very high success rate. Almost all of the pupae we received were able to turn into adults that then colonize those patches where we release them. However, if conditions were too hot, like they were in 2008 and 2010, you can see how many of the pupae did not make it until they were able to eclose and fly out of those contraptions. But starting in 2011, you can see we reared the pupae to adults indoors, and you can see we consistently had very high success rate with this method. Almost all of the pupae we received, we were able to basically convert into adults that were then released. And you'll also notice here that we stopped our accelerated colonization effort in 2015 because we really think that, now we have enough of these patches that are colonized that enough of these patches were occupied that butterflies are starting to colonize new areas on the preserve of their own. So now if we establish patches in any of these new areas, these butterflies can pretty much get there on their own. So we've stopped the accelerated colonization program in 2015. We could always start it up again if we need to, but right now we think that the butterflies can probably get there on their own to newly established patches. And we've seen evidence of that. Okay, so by this point, hopefully I've convinced you that we are putting a lot of effort into recovering these butterflies, but we'd like to know if our efforts are working. In particular, we want to know how our recovery efforts are affecting the population size. What kind of trends in population size are we seeing? How is the population size doing in terms of recovery criteria? And again, the recovery criteria here is we're looking for greater than 3,000 first or second brood adults in 4 out of 5 years. And also, how is our population distributed? Is most of the population concentrated in a few sites, or do they exist across the preserve? So to answer these questions, we've been monitoring abundance since 2007 using a method known as distance sampling. And this slide shows you here in blue, these show you our survey sites from last year. And you can see these survey sites are really just a subset of all the restored areas that are likely to contain butterflies. And so if we zoom in on these survey sites, you can see they're covered with a series of transects. And a single survey consists of our transects walking all of these transects across all of these sites all within the same day is considered one survey. And then field techs conduct nearly 30 surveys throughout the spring and summer of each year because we want to make sure we have many many surveys across the first and the second brood. Now remember, the quantity that we're interested in here is brood size. We want to know, which is, and again, brood size is the cumulative number of butterflies that emerge within a given flight period of that brood. And we estimate brood size using a two-step process. We start with the raw counts that we get from distance sampling, and then we adjust those accounts for imperfect detection to estimate abundance. So we go from raw counts from each survey to estimates of abundance for each survey. And we do that by accounting for imperfect detection. And what I mean by imperfect detection is that when we're walking along these transects, we know that we're not seeing all the butterflies out there. So for example, suppose on a particular day, we walked all the transects at a site and we counted 100 butterflies. Now suppose we know that we only detected half of the butterflies out there that were actually present. Then the estimate of the total number of butterflies that would actually be out there that day would have been 200 butterflies, right? It's going to be 100 divided by 0.5 if we had a 50% detection rate. So I'm not going to go into any more details on that. That's sort of the gist of what we're doing for this step. And that's the reason why we're using distance sampling methods, because that method allows us to estimate detection probability, which then that allows us to adjust our raw counts to get to abundance estimates. So that's the first step of this. The second step then is to come in and fit an insect population curve to those abundances. So this is why we want multiple surveys per brood. And so we can fit a curve to that brood. And there's parameters that control the shape of this curve. So we fit the curve that fits these points the best. And then the parameters, the estimates of those parameters that control the shape of that curve, some of those parameters give us useful biological properties, such as start of eclosion date, when do they start to, the adults start to emerge, length of the eclosion period, mean lifespan, and then the quantity here that we're after is the brood size. Remember, the brood size is the number of individuals that emerged over this entire period. Remember, individuals come out, they only live for 3 to 5 days. So the population across this period is constantly turning over. So the estimate that the quantity that we're after is how many butterflies emerged over this whole period. So I'm glossing over a lot of details here, but that's just really just to show you our general approach of how we go from walking through the field, counting butterflies, to getting an abundance of how many butterflies are in each brood. Okay, so how are the butterflies doing? So the first thing I want to remind you of is the amount of area that we survey has increased. Remember, as we restore pitch pine scrub oak barrens and then we release butterflies into those barrens, we would include those areas into our survey effort. So the amount of area that we've surveyed has increased over time. Now we're sort of maxed out at about 50 hectares. We really can't survey much more than that, even though we have more restored habitat than that. But so we've increased the amount of area we've been surveying over time. And now let's overlay the brood sizes. And so you can see since we started counting, the number of butterflies has increased. Up until the past few years, we've had a little bit of a decline. But overall, you can see our recovery efforts appear to be working. From where we started in 2007 to a few hundred individuals, to where we are now in the thousands. Now remember, the recovery threshold is 3,000. This is the federal and state recovery threshold. And we've exceeded this threshold for the first time. The second brood exceeded it for the first time in 2013, and then both broods have been over that since then. So at our particular population, which again, remember, is only one state recovery unit in New York State, and only one of the 3 recovery units that's needed within Glacial Lake Albany Federal Recovery Unit. And remember, there's 13 recovery units across the entire US. But in terms of our recovery unit, we have, for the last 7 years, have met the recovery criteria. And also keep in mind, so we are above this threshold of 3,000, but keep in mind that these abundances are just based on the survey sites. So these areas are, the estimates are based just on the areas that you see here in blue. There are butterflies at nearly all of these areas where we've planted lupine. We just really can't survey any more than we already are. So we can't survey these areas in green, but we know there's more butterflies out there. So the estimates I showed you here in this graph are very conservative because they're only based on the total of all of these blue areas. So there are many more butterflies out there than the estimates that I've come up with. And all the butterflies are not equally distributed among these survey sites. To give you some idea of the relative contribution of these different areas, we grouped sites into subpopulations. And the groupings were based primarily on their proximity to other sites. In general, if a site was within 200 meters of another one, they were considered to be the same subpopulation unless they were divided by a geographic barrier, such as a road. And here's the relative contributions of the different subpopulations to the broods in each year. So you can see clearly the butterflies are not equally distributed among the subpopulations. But the big takeaway, I think, from looking at this is to show that we've diversified the distribution of the population. We are getting our eggs into multiple baskets, so to speak, which is really important because you can imagine if we have all of our butterflies into one subpopulation and something bad happens to that one subpopulation, there's a good chance that that we can lose the entire species at the preserve. Now our population is diversified. We have them in lots of different places within the preserve. So now if anything happens in one particular subpopulation, we will have these other subpopulations that can act as a source of colonists to any individual subpopulation if something goes wrong at that subpopulation. Now, in addition to increasing the abundance of the butterflies by increasing the quantity of the habitat, we also seem to be increasing abundance by improving habitat quality, and that be seen here by seeing the increase in density of the butterflies in at least some of the subpopulations. So you can see there's fluctuations. There's always going to be fluctuations in insect populations, but you can see in a lot of these subpopulations we've increased the density of butterflies in those, which suggests that we've increased the quality of that habitat. Okay, so based on abundance, distribution, and density, we appear to have been successful in pulling our local population back away from the brink of extirpation. And we're going to continue to manage, we're going to continue to monitor the population, but we're also interested in understanding why were we successful? Like, what is a mechanistic relationship between management and Karner blue population size? And so the link between management and Karner blue population size is habitat. We manipulate habitat through management, and then the Karner blue population responds to those changes in habitat. So what we want to know is how is our management, how are our recovery efforts affecting habitat? And in particular, we want to know how is the management affecting the habitat characteristics most important to the Karners. So that we want to know, are we increasing lupin density? Do we have a diversity of nectar species out there that are well distributed throughout the preserve? Are we creating vegetation structure that is suitable for Karner blues? And so we're doing that by monitoring Karner blue habitat. And we're doing that, we're monitoring Karner blue habitat at 7 sites across the preserve. And these are also sites at which we're counting Karner blues. So at each site, we put out transects, multiple transects at each site, and then we collect data along those transects, and then we use those data to estimate the density of lupin. We determine the presence, absence, and distribution of high priority nectar species, and then we characterize the structural features of the vegetation. And then we revisit all of these transects, the same transects at these sites each year, and we collect the same data so that we can start to look at the trends of these different habitat components. So as an example, this is what these data look like. So here are 4 of the habitat components we monitor, and these data are from a site out by the landfill restoration. These areas that you see in different shades of green, these are represent the values of the different habitat indicators that we think we rank. This is the ranking of what we think of these different values take on in terms of are they poor, fair, good, or very good. And these are largely working hypotheses based on our current understanding of Karner blue habitat requirements as well as some preliminary modeling work. So for example, we started monitoring this site in 2015, and you can see the lupin density there has in the hundreds of thousands stems per hectare, and it's been increasing since then. These are all, this is much more lupin than we think the Karners need out there, so these are all in the very good category. So much so that you can't even see all the other categories are smushed here down near the 0. Summer nectar species is one of the nectar measures that we are monitoring. This has also been very good, suggesting that there's enough nectar species out there to provide a consistent supply of nectar for the second brood. Grass cover is an example of a vegetation structural characteristic that we're monitoring. So you can see when we started monitoring this site for the habitat. The grass cover started out a little low and increased and got into this middle area here where we think is best for grass cover, and then it looks like maybe the grass is starting to get a little bit too widespread, and now it's good, but not very good. And then finally, here's another example of a measure. This is an overstory canopy. This is another measure of vegetation structure. And you can see clearly, we currently we have too little canopy cover out there. And this is largely because there's just a lack of trees out at this particular restoration site. So you can see our canopy cover is in the poor category, because there's just really not much out there. So we're interested in collecting this information because eventually what we'd like to do is look at the relationship between management and these habitat metrics, and then look at the relationship between these habitat metrics and then the Karner numbers. And so I'm still pulling this information together. In particular, I'm still working on getting brood size estimates for individual sites. So I'm not yet ready to relate these habitat metrics to the Karner numbers, but I don't want to leave you hanging completely. So for now, we'll just take a quick look at how are these sites responding in terms of time since management. And you can think of time since management, this is sort of a surrogate for all the changes in the vegetation that occur as it regenerates after management. And this is just here to give you an idea of how quickly the vegetation responds following management. So here's a series of photos that were captured on a wildlife camera. This wildlife camera was put out on June 9th. This was 1 week. This is put out at Bivy, at our site called Bivy. This was 1 week after it happened. So this was essentially ash on June 2nd. This is 1 week in. So now as I'm gonna flip through these photos, here's a photo every 2 days for the next 2 weeks. And you can see, as I flip through these slides, hopefully you can appreciate just how quickly the vegetation regenerates. So that is just 3 weeks after, again, the site was burned pretty hot. That site was mostly ash. And so just in 3 weeks, you can see how quickly the vegetation has already regenerated. So keep that in mind as we think about time since management. So we manage, knocks back the vegetation, and then the vegetation grows back after management. So here's a graph of how the raw counts of Karner blues change following management at a site, Ellicat. So the black lines here are the raw counts for each of the broods. This is so each day that we survey is represented here. So like first brood, second brood, first brood, second brood. And the vertical lines indicate that the time that the unit was mowed or burned. So you can see this one in late 2013, it was mowed, it was burned shortly after that, probably a month or two after that. And then you can see following management, there was an increase in the raw numbers of butterflies. And then an increase, but that increase was temporary. It attenuated over the next 3 years. My guess is what we're seeing here is that the mowing and burning knocked back the scrub oak. It opened up the vegetation for lupine and other nectar species to flourish. But then after a couple of years, that scrub oak did grow back, and it probably started to crowd out and shade out the lupine and the nectar species. And so we started to see a decline in those numbers again. We also saw a similar response at this is at site Asphalt. But this one we saw a similar response just from mowing alone. And this is interesting because it could suggest if we want to increase Karner numbers at the site, we could just mow it. However, Neil's on the phone, and I want to make sure that I point out, it's important to note that mowing is not a long-term replacement for burning. Burning has effects on the ecosystem that cannot be duplicated with mechanical management, such as mowing. So mowing could provide a temporary boost, but again, it is not a long-term replacement for burning. We could mow temporarily, but eventually we need to get back in and return fire to those sites. However, just to give you an idea here, an increase following management is not always the case. So here you can see in Asphalt, this is where we mowed it. We got a temporary bump in the numbers. They declined. Then we came in 2017 and burned. Then we saw hardly any effect at all following that burn. And then you can see here is another site called Durgasitz. We mowed here in 2013, and we saw almost any effect, hardly any effect at all. So what this shows us is clearly we don't have all the answers. We still have a lot of work to do to figure out why mowing and burning increases populations on some sites and times, but then it doesn't at others. So there's more going on than just the mowing or burning, opening up the understory story and then letting the vegetation grow back afterwards. And so really what this, for me anyway, underscores the importance of really, we have to dig in and really look at understanding how management affects the habitat and then how habitat affects the Karner blues. So time since management can be helpful, but it clearly is not giving us all the answers. Okay, then looks like we're about out of time, but what I want to leave you with here is we still have a lot of work to do, obviously, to figure out exactly how our management can best help the butterfly. But I think we know in a general sense that what we're doing is working, or at least we know it's working for now. What will happen as we look into the future? In particular, I'm thinking about climate change. How is that going to affect the Karners? Can we expect our population to increase? Will it remain stable? Is it going to decrease? And I'm not talking next year or anytime in the near term. I'm really talking here about 50 years from now. What is it going to look like for the Karners? And first of all, we know that the climate is changing, and we can see that locally. These graphs are based on data from the Albany airport from 1938 to present time. And these graphs show that the average daily maximum temperature, it shows the average daily maximum temperature for each season is changing over time. And the slopes are significant for spring and for winter. So the slope here is 0.02. So what that means for every 50 years is going to lead to an increase of 1 degree Celsius or about 2 degrees Fahrenheit. On average, the daily maximum temperatures will increase in the spring and in the winter. Here's the seasonal average of daily minimum temperatures. So you can see this is, you can see the daily minimum temperature increasing across all seasons. All of these are statistically significant. So how might these changes in temperature affect Karner blues at the Albany Pine Bush Preserve? Well, we're not doing that research here, but other scientists have been looking into this. The University of Notre Dame maintains a captive colony of individuals that were captured from the Indiana Dunes National Lakeshore. And what they did there with that captive group of butterflies is they exposed that group of butterflies to 4 temperature treatments. The 0 degrees temperature treatments was just the average temperature regime, the historical temperature regime from 1952 to 1989 at the Indiana Dunes. And then they also exposed them to temperatures on average 2, 4, and 6 degrees above that historical temperature regime. So in 2011, they found that higher temperatures induced more broods. So you can see the historical temperature regime here, +0, that gave the typical 2 broods, is what you typically see. But now as they upped it to +2 degrees and 4 degrees, they got an extra 3rd brood. And then when they went to 6 degrees, they actually got a 4th brood. However, in that 4th brood, none of those individuals pupated. I'm guessing they didn't have enough time because 4 broods is a lot to pack into 1 year. A similar pattern, they repeated the experiment again in 2012. A similar pattern was found where they saw an increase in the number of broods, but it became apparent here just at the +6. They didn't see it in the +2 and 0.4, but at 6, they still got 4 broods again. So these results suggest that warmer temperatures are going to lead to increased volatilism, and that just means that we're going to have, potentially, you can have more broods per year as the temperatures increase. Here are some results from the same experiment. Here we're looking at the mass of the adults from each of the broods in each of the temperature treatments. So each of these different colors are the different temperature treatments. And here are the adult masses for the 1st, 2nd, and 3rd brood. And so you can see there is a decreasing, there's a trend in decreasing adult mass with successive broods. And it's strongest for this +6, and that's really prominent here in 2012. You can see the broods, the adult mass size really decreased when in the +6 temperature treatment. So these 2 pieces of information taken together, if the warmer temperatures lead to more broods and then more broods lead to smaller adults, and you combine that with the knowledge that smaller adults tend to be less fecund, meaning they will have fewer eggs, that's going to potentially lead to less eggs leading into the 1st brood in the following year. So that's not necessarily a good thing that we're getting more broods and smaller adults that are laying less eggs that are then have to overwinter and start the 1st brood in the following year. Thus, climate change is probably going to be detrimental to the population size of the Karner blue. In addition to climate, in addition to changes in abundance, there's also going to be changes in distribution. Thermal tolerance often determines the geographic ranges of insects. So if you alter the climatic conditions, the insect ranges are probably going to change. So to better understand the species' current distribution and how that distribution might change under a climate change, a group of scientists from Indiana Dunes National Lakeshore used a climate envelope model. And this model describes the current distribution by relating the known occurrences of the butterflies to bioclimatic variables, and then they forecast how do those bioclimatic variables change based on the changing climate. And so this model forecasts almost a complete extirpation from the current range, with the exception of maybe central Wisconsin. They're still hanging on there, but by and large, the range is going to move completely north, and completely is going to be very little overlap with where they're currently are by 2080, is what the model's forecasting. And just a couple more things here. In addition to directly affecting the Karner blue population, climate change is also probably going to affect the very processes and methods that we are using to try to achieve our conservation objectives. For example, climate change may alter the effectiveness of fire as a management tool, or it may lead to unsuitable locations for habitat creation. So for example, if climate conditions push the range northwards and now all of a sudden we need to create habitat where there's a whole bunch of human development, we might not be able to create new habitat into those new locations. Currently, our primary management goal is to try to restore the vegetation to its historical conditions. But in the face of climate change, we may have to modify our goals to adjust to climate change. If we keep trying to push the ecosystem in the direction that climate change will no longer sustain, we may be shoveling sand against the tide. And then because climate change is going to bring conditions that differ from the past, it really is going to challenge our current preservation-based management objectives. Like I said, we're trying to preserve it sort of in its current state, but really that might not be possible moving into the future, and we might have to consider the implications of novel communities and ecosystems. Okay, so I'll leave it there. Just to conclude, I'd like to say that I think in a general sense that our management is working. I think removing black locust, doing things like removing black locust, thinning out scrub oak, doing prescribed burns, planting in lupine and other native species. I think that's obviously, we're having a positive response from those recovery efforts. I think you can see that here in our population trends. And that's great news. Like I said, we've pulled the butterfly back away from the brink of local extinction. However, our work is not done. We still have a lot of work to do. In particular, like I said, we're interested in understanding how management affects habitat, and then how that habitat then affects population size. And understanding this causal chain will be really important to us now because it will help us better manage for Karner blues, but it's going to be especially important, I think, as we head into an uncertain future, especially with regard to climate change. But I do think for now, our population is doing well, and I think it puts us in a relatively good position to deal with whatever the future may bring. So with that, I would just like to quickly acknowledge the Environmental Protection Fund, where we get our funding from for all the work we do as a commission across the entire preserve. Fish and Wildlife Service supports a lot of the work that we do. The National Fire Plan grants gives us a lot of helps pay for our management and fires. And then I would be remiss if I did not mention our many field technicians, interns, and volunteers who have collected. They're the real workhorse here. They're the people that are really collecting all of the data over the years, because it's the three of us, Dylan, me, and our conservation director, Neil Gifford, we can't keep up with all the data that needs to be collected. So our field technicians really do a great job at and really collect a lot of the data that we need. All right, thanks. With that, I can take some questions.
All right, thank you, Steve.
Sure.
So we do have some questions. If anybody has any other questions, feel free to type them either into the Q&A box, or you can put them in the chat box. I'll check both spots. To start with, Michael Lesser says, can the Karners be reintroduced in new areas? If so, how? I have heard that they will be trying to reintroduce in the Moreau Lake State Park area.
Yeah, that's a good question. And that's what I was going to point to, is the Moreau Lake State Park. They're thinking about potentially putting in another, making that a new New York State recovery unit up there. And so I think the first thing they need to do is try to get the habitat favorable first. And so that's going to probably require a lot of the, similar to what we're doing here, which is making sure that the canopy is open, making sure that they have certainly lupin and shrub components. They basically have to make sure that all of the vegetation components are present, and then you can think about releasing individuals, and you probably need to release enough individuals to get a large enough population in order for it to get it to become established. But sure, it is absolutely possible. I think we've shown that here, all the different places where we've gone in, they were empty, we've released them, and they're successfully hanging on in a lot of the places where we've released them. So yes, I think that can and very well may happen up there.
All right, and kind of similar on that grain, Margie asks, how about the Rome sand plains? Is there a population of butterflies there as well?
I do not think they're currently there, and I don't know if Dylan or Neil can jump in, but I don't think that the habitat there is being managed well enough for them to think about them being released there. I mean, certainly if it was, it's historically, yes, but I don't know if they have the work done now to consider releasing that, or if it's even on the horizon of having that work done. Potentially, yes, but I don't know that they're doing any work there. Dylan, have you heard anything?
I haven't heard anything. I know it is occupied by frosted elfin, which is another SGCN species that's also a lupin feeder. And is currently being evaluated by the Fish and Wildlife Service for possible listing, but I don't know that there's any plans.
Yeah, my guess is they occurred there because like there's lupin there, and so they probably just got locally extirpated. And if we're the closest source population, they're not going to get there on their own. We're going to have to be brought in.
Yeah. Okay. Someone asked, how far will a Karner blue butterfly travel from where it hatched from its egg during its lifetime?
Well, that's why we define the subpopulation with the 200 meter. If it's within 200 meters of an occupied patch, we consider it to sort of be within the same subpopulation. So on average, that's what we think they move, is up to 200 meters. Obviously, we have seen some, I think in the mark recapture study that was done years ago, I think they did find some moving up to 1 kilometer, but that's relatively unusual. So I think 200 meters is, within 200 meters is probably closer to what you can expect.
All right. Neil did chime in about the Rome sand plains. He said it's a potential New York State recovery area, and it did historically have Karner blues. Okay. Someone asked, do you have data on how mowing and burning affects biodiversity of all species in addition to the Karner blue?
Yes and no. I mean, we have biodiversity is sort of it's a catch-all term, but we know I talked about mostly what its effects on Karners, but we're bringing back the ecosystem in the northeastern interior pine barrens ecosystem, the inland pitch pine scrub oak barrens. We're not bringing this just back for the Karner blues. There's a lot of other those I mentioned, those 78 SGCNs. There's a lot of other wildlife species that depend upon this, and they do some like very, very much like the same conditions that the Karner blues do. Others do not. For example, when we mow and burn within a year, that's good for Karners, but it probably is going to take 4 or 5 years before someone like a prairie warbler, until there's going to be enough shrub cover for prairie warblers to move in there, or other shrub-loving birds like towhees and thrashers, which is why I didn't get into it. But something that we think about is how are we, every time we treat something, that time since management, right? We're setting back succession, and we're sort of setting back the clock. And so for that particular patch, it's going to be good for some species that need a year, and then a year from there it'll be 2, 3, and 4 years, and 5 years out. And we have a mosaic kind of going on in the landscape. We're burning and mowing all different places and all across the landscape. And so we have different patches and different ages at all different times. And so the idea here is that most of the species in this landscape have sort of evolved with this, we call it a shifting mosaic. All these different stages of succession they're moving in and out of different age classes across the landscape. And so these things are constantly kind of shifting around, chasing the sort of habitat that works best for them. And so within a particular site, if we manage for something like that, it'll be good for Karners immediately, but not necessarily good for everything else. It'll be good for a lot of things that use the same habitat as Karner, but not everything else. But on the landscape, across the landscape, yes, we're very concerned about how we're affecting all the different species, and we're trying to maintain all the different age classes and all the different habitats that we need for all of these different species that use these inland pitch pine scrub oak barrens communities. I should have just pulled up a picture. It probably would have been easier to show you about our shifting mosaic with a picture instead of me trying to ramble on about it.
Let's see, Scott, maybe if you could go back to your chart of the population sizes through the years. Scott is wondering if you can please describe and discuss why you think there was the huge population boom in 2014 and 2015, followed by that subsequent decline, if you have any.
Yeah, that's a good question. So we think a lot of this was driven by, we had one site that this year really for 2 years, one site really carried this population up to this peak. And it was just, we restored the site. We had a ton of lupin in the site. The site sort of went crazy for a couple of years, and then it sort of settled down. And so I think this peak was driven a lot by that. I think it was also driven by favorable winters. You see, typically we see kind of the seesaw pattern where we have our second brood lays eggs. A lot of those eggs die, and then the first brood recovers to the second brood, and then up and down, up and down, up and down. So I think it was probably a lucky confluence of we had one really good site that came online in these 2 years, and then probably with good winters drove this up. And then this is probably more typical here. We're just going to see ups and downs based on the different management that we're doing. I should add, I don't know if any of you guys saw the press release in the paper, but we I think we've had another good year of favorable weather in 2020, and we had another couple sites that we had just treated recently that caused the population of those sites to spike. And so my guess is we're low here, but my guess is we're going to be off the chart, literally off the chart next year when we come up with the population estimates. We don't know that for sure until we collect all the data, but it's looking like right now that the population is going to come surging back up. But as you can see with these types of populations, they're very flashy. You can get explosive growth, and then you can get crashes. And so the idea here is that even after we crash, hopefully we're still above our 3,000. So the populations are going to fluctuate. This, I think, was due to, like I said, we had a good site, and some favorable winters led to the population growing, but then we probably had some bad winters, which then crashed the population. They recover a little bit each year, but then they go back down.
Okay, I'm going to go to Neil's question because I feel like it might build off of that a little bit. He says, how do non-habitat environmental factors influence population growth?
Yes, so with insects, our insects don't do well. They don't buffer well against temperature extremes, right? They're out there. They're not like us or deer or whatever. They can eat. They can stay warm, or if there's droughts or lack of food, those types of species can weather sort of bad environmental conditions in terms of temperature or lack of food. Insects are really vulnerable to we call them density independent effects. Like, it doesn't matter how big the population is. If a cold snap comes in, it's just going to wipe them all out. Or if it gets too hot, they're just going to cook. They have very little ability to buffer against the elements, and so that can cause them to populations to crash if conditions become unfavorable. But the other side of that is if conditions are favorable, if they are having good weather, typically these insects, they have very large clutch sizes. Karners lay about 300 eggs. So if each Karner lays 300 eggs and the conditions are good, the population can grow very quickly, can grow very large very quickly. And I think that's what we're sort of seeing this year in 2020, which doesn't show up on this graph.
All right, and in a similar vein, Darwin says, although we have had relatively mild winters in recent years, what happens to brood one populations following a winter that is especially cold with deep snow depth from, say, December through mid-April?
What we think of if the snow is deep, that actually buffers, and if it gets really cold, if it gets really cold outside, if they're blanketed, if they're under a nice blanket of snow, I forget exactly if it's a foot of snow or what it is. Oftentimes at the interface between the snowpack and the ground where the eggs are, that'll still be close to freezing, like 32 degrees, and it'll be sort of constant 32 degrees, no matter what the temperature is doing outside. So even if it gets really cold or really warm, the snow acts as an insulating barrier that will keep the eggs near 32 degrees, just above freezing. Now, without that snow, that's when it's exposed to the really cold snaps. And it's also, the fluctuating temperature also isn't really good for the eggs overwintering either. If they're getting really cold and then warm, that also leads to increased mortality. So yeah, and I would say that we haven't really had really great snow cover over the last stretch of years. If we get snow at all, we might get a foot of snow and it seems like it's melted 2 weeks later, and then those eggs are then exposed again. So for snow cover, you need consistent snow cover across the entire winter to really keep those eggs protected, and that will help reduce winter mortality.
All right. Okay, Dick was wondering, what is the ratio of survey site size to restored area?
I think I did that once. I think it was about a third, maybe. So I know what you want to do. You just want to take our population estimates and multiply them by 3, but it's not quite that easy, right? You need to account, the habitat is not the same everywhere. Otherwise, I just would have done that. If we looked across all those things, some are grassy, some are closer to thickets, some are closer to pitch pine oak forests. So you need to figure out what the density is in those different types of habitat types, and then try to project that to figure out what proportion of those different areas are of those habitat types, and project it out that way. And I feel like I'd be doing a lot of hand waving to come up with a number that might not be very reliable. So yeah, and so I'm not comfortable just doing the back of the envelope multiplied by 3. You can do that if you want, but I'm not going to recommend that approach. But okay. It is clearly higher. There is more habitat out there than we are currently surveying in, and I was only comfortable making inference to the areas that we were surveying. But we know that our populationists are conservative.
And we do regularly visit some of the sites that we're not surveying with distance just to confirm presence.
Yeah, that's how we know. Like usually every 5 years we'll go out and look to make sure that those sites are still occupied. And for the most part, there's butterflies hanging on into all these areas.
All right, Richard wants to know if birds eat the pupae.
Yeah, this actually question just came up Tuesday, and Dylan's, I think, answer was no. They're kind of small and hard to find. And I think a bird would probably spend more energy probably trying to poke around and find them. Sure, if there's one poking around and it's right in their face, they might eat that. But my guess is they're going to have a lot better luck trying to pick caterpillars off of oak leaves than trying to pick small, well camouflaged larvae off of lupine. So possibly, but probably not. Probably insect predators are probably a bigger source.
All right. Last call for questions. I've got one more. April was wondering, how many eggs can a female lay before she dies? And do they lay one egg or multiple eggs on each wild blue lupine?
Oh, well, we know from the lab, from New Hampshire Department of Fish and Game, when we were doing the captive rearing, we would send the females over and they would collect that information. And I think they could lay up to 300 eggs. Am I getting that right? Neil can chime in. He just looked at those numbers. I think like maybe 100 up to 300. And they do lay them individually, don't they? Dylan, they don't?
Yeah, Neil actually just commented 1 egg per lupin stem based on field studies.
Yeah, that's right. I should know that. I did know that at one point. I forgot, but yes. So 1 per stem. So it's not like some of them, like our inland barrens buck moth, will lay a bunch of them in a clump. They lay individual eggs on individual stems.
Neil also said the average for the captive rearing, the number of eggs laid, the average was 160. The max was 313.
Okay. Yeah, like I know he just had those numbers recently.
All right, let me just double check, make sure I got everything. I think I did. All right, well, thank you very, very much, Steve. That was a very dense, dense presentation with a lot of great information. We really appreciate you talking to us about it tonight.
Yes, thank you everybody for tuning in.
Yes, thank you everyone so much. And have a good night.